Thanks, Emma, for that introduction. Today, as Emma mentioned, I will be talking about a metadata package that we made to share wastewater microbial data. Let me grab this. All right, so a quick overview of this contextual data spec. Our goals with this specification were to standardize how wastewater metadata is stored, to provide recommendations around how you should capture this metadata, while maintaining relevance for a wide set of contexts. So viruses, bacteria, AMR, parasitic worms, etc. We also wanted to have interoperability between our standard and other currently existing standards, and to do all of this in a co-creation effort with collaborators from different contexts. So we are delivering as part of that a spreadsheet template that labs can actually use to fill out the different metadata fields, a field reference guide that's very helpful for explaining for each term how you should be using it, what its definition is, some guidance around it. All of them have ontologies. As well as we include mapping files between our fields and fields from other data specs, a standard operating procedure where we outline some of the ethical or privacy considerations that should be taken into consideration. And then also we will have a public repository that will allow not only us at PHA4GE to keep updating the spec, but also for anyone in the wider community to make suggestions and help us edit and revise the specification. So talking now about wastewater surveillance. Wastewater surveillance started under 100 years ago with polio, and that polio environmental surveillance still continues to this day. So on the left, you can see a mapping of surveillance in Nigeria in the early 2010s for polio. And here on the right, you see a map of the US with currently operating sites for SARS-CoV-2. So just during this pandemic in particular, there was a huge amount of investment in not just infrastructure, looking at how many sites we have, not just in the US, but around the world, but also in the technology we need to do and analyze this surveillance. Now, the next step after having all of this investment in infrastructure and in technology is to then move on to what are our next potential applications. So wastewater surveillance can be applied for, as I mentioned before, a wide range of parasites or pathogens, viruses, parasitic worms, bacteria, antimicrobial resistance. And it can also be leveraged not just for looking for one particular pathogen, but for within a single sample, looking for multiple pathogens. In addition, you can do this surveillance in a wide range of systems. Open sewage systems, closed sewage systems, as well as open, possibly contaminated surface waters. So when we're talking about all these different systems, viruses, bacteria, open, closed, just a contaminated body of water, this is where having standards around metadata becomes very important to make sure that we're speaking a shared language. So looking now at the spec that we are still in the process of creating, one thing that was very important to us in creating this spec was to follow FAIR guidelines. FAIR standing for findability, accessibility, interoperability, and reusability. So here on the left, you can see how this spec breaks down on a couple of different axes. Starting with that second column, you'll see that approximately 60% of the terms in our spec either have an enumerated pick list, meaning that when you are filling in that term, you pick from a list of potential values, or the format for that term is very structured. So it's a date, it's a time, integer, or decimal. This allows for machine readability of the data that you then share with potential collaborators. In addition, we have over 10 fields that store identifiers for where you have shared the data and metadata in relevant global databases. This is to allow for accessibility. Further, approximately 40% of the terms in our spec map to current other global standards, and we will, as more standards come to be used, we can also continue to make these mappings between our standard and other global wastewater standards. In addition, the spec uses about 20% of the terms from our spec are terms that are actually used in the phage SARS-CoV-2 spec. And this implies not only that that spec was reusable, but that our spec will also be reusable. And I imagine this in particular being relevant for other environmental samples, as well as other metagenomic sequencing approaches, where some of the terms that we are coining now, and 80% of them we are coining now, so that's a decent amount of effort, will now decrease how much effort it takes to create future standards. We also, within this spec, mark which fields are required, recommended, versus optional. That's that third column in green. And I want to mention that only 6% of the terms in this spec are actually required, which is really low. Part of why we have over 90% of the terms in the spec that aren't required is to operationalize how you would store that data. So if you, for example, in your lab always measure pH or turbidity, how would you record that? What's the best practice there? We're not trying to say that everyone should measure 20 physicochemical metrics for every single wastewater sample that they have. But we want to make sure that if you are doing that, that there's a standardized approach to how you collect it, record it, and then share it. Finally, we also split these terms into 6 different categories, and that just makes the spec more navigable. So I'll introduce those 6 categories now. The first one is database identifiers, which denotes where your sample has been shared. The next is sample collection and processing. This records all of the processes from before sample collection through to extraction. We also have environmental conditions and measurements, which are internal or external properties of your sample. Sequence information describes the procedures that you use to generate reads for your wastewater sample. Bioinformatics and QC metrics describe the processing of raw sequencing data and how you analyzed this raw and processed data. And finally, pathogen diagnostic testing is which pathogens you detected in your sample prior to any sequencing efforts. So to go quickly one by one through these categories, for database identifiers, a lot of the fields are just what's the accession number for different databases where you might have shared this sample. But in addition, we have the site that you collected the sample from, as well as the event during which you collected that sample. So this helps you connect your sample to related samples across time and also across space. And then you can also see this coloring. This is that required, recommended versus optional categorization that we have. So green is required, yellow is recommended, and gray is optional. Moving to sample collection and processing, as I mentioned before, this is any process from before collection through to extraction. So that includes why did you choose to sample here in the first place? Why this sample? Where was it taken? What was the collection method, extraction method, and a number of other terms. Environmental conditions and measurements can be split into 4 types of metrics. One is human-related metrics, such as the population that contributed to your sewer shed. Others are weather-related, such as the sampling weather, or how much precipitation there was in the 24 hours prior to collection. We also have physicochemical metrics like turbidity, and then finally we have wastewater quality metrics like total solids and fecal coliform. For sequence information, this is describing the procedures that generate reads, and here we felt it was very important to incorporate not just terms for amplicon-based sequencing, like what was used for SARS-CoV-2, but also metagenomic-based sequencing. So if you look at those 2 terms that are in italics, those are terms that are relevant for one or the other. So amplicon PCR primer scheme, relevant for amplicon-based sequencing. Genomic enrichment methods are relevant for metagenomic-based sequencing. And so in addition to including these terms in order to make this spec pathogen agnostic, we are also going to narrow down this spec and provide different templates of how you should use this spec for different pathogens or for different sequencing approaches. Bioinformatics and QC metrics is how you process your raw sequencing data and analyses of that raw and processed data. So what were some quality control issues you might have found? What were the total number of reads, et cetera? And then finally, pathogen diagnostic testing is again where we try to make this spec pathogen agnostic. So we're not assuming that you're doing PCR-based testing. You also might be doing culture-based testing, CRISPR-based testing. And so we wanted to have terms that could encompass that whole range of potential methodologies. So this spec, actually appears it got cut off, so I apologize. There's supposed to be an extension that goes down below the floor. But our spec is meant to be succinct and comprehensive in how it describes different types of wastewater setups. So with these 3 fields, you can describe different setups. So for example, if you have treated effluent that's coming from a wastewater treatment plant, and if this plant receives both wastewater and also runoff, with these 3 fields, all of which have pick lists, so you're not entering these yourself, you're choosing amongst terms that have been pre-established, you can describe that it came from a wastewater treatment plant, that it's effluent as opposed to raw wastewater, and that this is a combined sewer system, meaning it has runoff as well. And there are a couple of other examples that if you want to see, you can ask me for it later. So just to end, because I'm actually past time, there's been a lot of investment in the technology and the infrastructure for wastewater. Data standards are what we need to bridge the gap between this technology and infrastructure and the actual public health implementations of this data. So as wastewater surveillance and genomic surveillance more broadly continue to develop, data standards is not only a key area where we as a community can work together, but it's also an essential piece of the puzzle for making sure that our public health systems are systematic, scalable, interoperable, and impactful. So thank you to everyone who's contributed. My apologies if you're not listed. And there's my email if you want to be involved.